Assalamu alaikum. So now you know amino acids and you know their classes, the different types of amino acids. So in the following three lectures, we will talk about how amino acids assemble into peptides and proteins, and how proteins have their three-dimensional structure. And then we will connect the structure of specific proteins into function. You know, how these are the beautiful lectures in proteins, in my opinion. Why is it that this protein has this specific amino acid in this specific position? Okay, so we're going to link structure of proteins to their function. So let's start with peptides and how amino acids assemble into peptides. The lecture is the main resource whenever you want to study, but you can also refer to Campbell's biochemistry textbook, chapter 3. It includes some nice review of peptide and protein structure. So let's talk about how peptides are formed, okay? So before we continue, let's agree on a number of terms or definitions. Okay, so if I say an amino acid, I'm talking about one single amino acid molecule, like glycine, tryptophan, arginine, whatever. Now, if I say an amino acid residue, it means it's an amino acid that is part of a larger structure like peptide or polypeptide or protein. Just like saying glucose and glucose residue. Glucose is just the molecule itself. Glucose residue is basically glucose that is part of a larger structure like glycogen and starch. If I say dipeptide, it means it is two amino acids that are linked to each other. If I say tripeptide, here I'm talking about three amino acids, and so on. Now, if I say oligopeptide, it's basically a short chain of 20 to 30 amino acids. So the number is really not definite. So it can be like 10 amino acids, 11 amino acids. That's an oligopeptide. Okay. Now, if I say polypeptide, it's larger. Okay, more than 30, let's say. So it's all, you know, relative. It's not something that is it's not like there's a certain line that divides an oligopeptide from a polypeptide. So it's all relative. So a polypeptide is basically a larger stretch of amino acids connected to each other. If I say protein now protein is a polypeptide. Okay, the difference is that a protein has a specific three-dimensional structure and it has a function, a defined function. A polypeptide is just a stretch of amino acids, large number of amino acids connected to each other, okay, with no specific three-dimensional structure. But when a polypeptide folds and it forms a three-dimensional structure, now that's a protein. So the average molecular weight of an amino acid is about 110 grams. That's the molecular weight. Now, grams per mole, that is. So usually the molecular weight of most proteins is about 5,500 to about 220,000 grams per mole. So they can be really huge. So if I say we have an amino, so I can say that we have a 10,000 molecular weight protein. It means that it has a mass of 10,000 daltons, or we say 10 kilodaltons. Okay, so you can sort of like estimate the size of a protein by knowing the number of amino acids that make up this particular protein. When two amino acids are condensed to each other, forming a peptide in organic chemistry, is known as the amide peptide. Okay, so this is the amide peptide, C-double-bond-O, and the C is linked covalently to a nitrogen, and nitrogen, there is a hydrogen. This is the amide bond. Now in biochemistry, that's in organic chemistry. Now in biochemistry, when we are talking about protein structure or polypeptides or peptides, it's referred to as a peptide bond or peptide linkage. Okay, and it's formed via a condensation reaction. So condensation because you have release of water when you have the formation of this covalent bond between the carbonyl group and the amino group right here. Now, there are certain features for peptide bonds. One of them is that it has a resonance structure, meaning that you have a conversion of a double bond. You have two structures right here. So normally you have this carbonyl group right here, C double bond O. Now you can have these two electrons right here within the double bond moving onto the oxygen, okay, and that results in the formation of a double bond between the carbon and nitrogen right here. So you have this resonance structure. So the peptide bond keeps on changing or transforming from this form right here to this form right there, and that results in the formation of the appearance of a negative charge on oxygen and a positive charge on nitrogen. Now, because of this double bond characteristic of the peptide bond, it makes the bond itself, or the group right here, the carbonyl and the amide right here, it makes it planar, meaning that it is flat. So this is a flat structure. This is a peptide bond right here. That's the carbon, that's the nitrogen. Okay. Now it makes the bond itself planar again, or flat. It's also charged, like I said, and because it has a double bond characteristic, the bond itself is rigid and it cannot rotate. Okay, so the bond right here cannot rotate. The bond right here cannot rotate, and notice that it is flat as well. Okay, but the bonds that can rotate are the bonds within the amino acid itself. So this bond right here cannot rotate, but the bond between the central carbon of the amino acid with the carbonyl group and the central carbon with the amino group, okay, these two can rotate, and these two are the ones that give a protein a specific three-dimensional structure. Something else is that the peptide bond makes the peptide having a zigzag structure. So zigzag, zigzag, zigzag. Okay, so so you have this zigzag structure right here. And finally, because of the presence of these groups right here, the carbonyl and the amide group, and because of the presence of partial negative charge on oxygen and a partial positive charge on nitrogen, because of that, the peptide bond, the groups that make up the peptide bond can form hydrogen bonding. So right here, for example, that's the carbonyl right here. That's the carbonyl right here, and this is the amide, and you can have the formation of this hydrogen bonding between these groups. Now, remember that certain amino acids, the ones that are polar charged amino acids, they can also form non-covalent interactions that are important in the formation of a three-dimensional structure of a protein. Now, the other thing here as well is that, again, you can have this rotation right here, okay, around the central carbon, okay? So these groups can rotate, as you can see here. But notice as well that as amino acids rotate, you can have a repulsion formed between the different groups. Okay, and you can see this in these lines, and that's why this is really important in the formation of a three-dimensional structure of proteins, because of this repulsion that is formed between the two groups. So groups would have specific distances between them in order to avoid having this repulsion between them. Okay, and that's what helps, or what helps in making up the three-dimensional structure of a protein. Now, something else about peptides and peptide bonds is that because of the presence of this linkage between the different amino acids, and it's continuous, you can have a backbone. And the backbone is basically the amide bond, the nitrogen, that is, okay, the central carbon, and the carbonyl group. And then you have the nitrogen, again, the amide group, the central carbon, the carbonyl, and so on. This is the backbone of a peptide. And in addition to that, you have side chains as well. And side chains are basically the OR groups that define amino acids. Okay, so here's a side chain, another side chain, a third side chain, and so on. Now, something else about peptides is that the OR groups are in the trans orientation. In other words, you have an R group that goes, let's say, downward, the one next to it, or the amino acid that is next to it has its R group going upward, okay? And then the following amino acid has an R group that is going downward, upward, downward. Again, this gives sort of like the zigzag structure of amino acids, or that's an outcome of the zigzag structure of amino acids. Okay. Something else as well is that a peptide has a polarity, that is, two opposite ends. So the first end is it's known as the N-terminus. Okay, the N-terminus of a peptide or a protein. And the other end is the carboxyl or carboxy terminus. Okay, so you have two opposite ends because one end is positively charged and the other end is negatively charged. Now, the end that exists at the N-terminus, okay, that has the amino group is amino acid number 1. So that's amino acid number 1, then amino acid number 2, then amino acid number 3, and here we have the carboxy terminus, that is, it has the last amino acid that is added when we form a peptide bond between two amino acids. So, okay, so whenever we want to add an amino acid, we add it to the carboxy terminus, not to the amino terminus. Just like DNA having a directionality going from 5' to 3', you also have the same thing with proteins going from the N terminus, that's the head, that's the start of the peptide, all the way to the carboxy terminus. That's the last amino acid that is added. Now, so like I said, whenever you have the formation of a peptide bond, proteins would tend to have the amino acids in the trans orientation. That is, you have the R group going right here, going upward, and then the one next to it, the R group would go downward. Okay, that prevents the formation of a repulsion between the two groups right here. Now, a difference is whenever you have a proline, and there's another difference, by the way, that I forgot to mention in the previous slide, the one before that, and that is, I said that peptide bonds, you can have the formation of hydrogen bonds between the groups that make up the peptide bond, except for proline. And the reason is that proline does not have an amide group, because the nitrogen proline is occupied by 3 covalent bonds. So it doesn't have a hydrogen that it can donate in hydrogen bonding. Okay. Now something else about proline is that it doesn't really have any preference for trans or cis orientation. So cis is basically when you have the 2 R groups or being oriented in the same direction. Okay, for example, right here they're going downward. Now, if both of them go downward, they would be, the 2 R groups would be close to each other, resulting in a repulsion between them, and that's why you have the R group, one going up and the other going down. Okay, that prevents the repulsive forces, the formation of these repulsive forces. Okay, that's why the amino acids prefer to be in the trans orientation. But for proline, there is repulsion either way. So there's really no preference for trans over cis. Okay, so that's another difference between that's another information about proline. Okay, so let's talk about a number of peptides. Now the thing is, and their functions. Now the thing is, whenever you have a peptide, many of these peptides really do not have any function. Okay, you have to have them form a specific 3-dimensional structure, and usually peptides are too small to have a specific structure. But there are certain peptides, really short stretches of amino acids, that do have specific functions, and we'll talk about these peptides and their function. So one of them is known as carnosine. Carnosine is a dipeptide. A dipeptide of 2 amino acids, and these are alanine and histidine. Except that this alanine is actually in the beta orientation, or the beta form. So what do we mean by that? What we mean by that is that whenever you have an alpha alanine, this is the alanine that we know of. So you have the central carbon with the carboxyl group, and you have the amino group right here, and that's the R group. And the R group of alanine is a methyl group. Now what happens is that with alanine, you can have the formation of what is known as a beta alanine, where you have the amino group connected to the beta carbon. Now this is the alpha carbon right here. Okay, that's the central carbon, that's the beta carbon, that's carbon number 2 in the R group. Okay, now you have the amino group connected to the R group, the methyl group, the beta carbon. Okay, so this is carnosine. Now this carnosine is important in muscles and in brain tissues as well, whereby it protects cells from reactive oxygen species and peroxides, and they play a role in contraction of muscles as well. So you can look at this structure again. Okay, so this is how it looks like, the B-alanine-L-histidine right here. Okay. Another peptide that is important is a tripeptide known as glutathione. Glutathione is made of 3 amino acids, and these are glutamate, cysteine, and glycine. Okay. Notice that the glutamate is also known as the gamma glutamate. Now, gluta, by the way, means 5 carbons. Okay, so this is an amino acid that has 5 carbons. Now, this is the normal structure of glutamate. Okay, the usual structure of glutamate. So right here you have the central carbon, the alpha carbon right here. Okay, and what you have in here is the amino group, and then this is the carboxyl group. Okay, so what happens, so this is the alpha, this is beta, and here you have the gamma carbon right here. And what happens is that the peptide bond is formed between this carboxyl group right here, okay, and the amino group of the cysteine. Okay, so it's looking at basically, so that's why it's known. This is known as the gamma glutamyl Okay, so gamma glutamyl cysteine glycine. Okay. The main function of glutathione is to protect our cells from oxidizing agents, that is, reactive oxygen species, free radicals, and so on. So radicals, again, these are molecules or groups that are missing a carbon, and they are very reactive. They want an electron from any group. So what they do is that they attack other groups and they extract this electron from them. So they get reduced and the other molecules get oxidized, and that's why they're known as oxidizing agents, because they oxidize other molecules. Now, when they oxidize other molecules, these molecules are damaged, like DNA, lipids in the plasma membrane, sugars, proteins, and so on. So if DNA is damaged, well, that's a problem. That would cause the creation of a mutation. If lipids in the plasma membrane are damaged, well, that's a problem because the membrane is damaged and cells may die. So what glutathione does is that it commits suicide. So what it does is that it puts itself in front of the oxidizing molecule or the reactive oxygen species, and what it does is that it says, take the electron from me. So it acts more like Superman, sort of, except that Superman doesn't die. Well, glutathione does. So what happens is that the sulfhydryl group of cysteine is in glutathione, it is reduced and it gets oxidized. So you have the formation of two molecules of glutathione connected to each other via a disulfide bond. So the molecule gets oxidized and the other molecules, the active molecule, would be reduced. Now, this oxidized form of glutathione, by the way, can revert to the reduced form, okay, with enzymatically as well. Another important peptide is known as enkephalins, and these enkephalins are pentapeptides that exist in brain tissues, and they function as analgesics, so they relieve pain physiologically. Now, there are two forms of enkephalins, and they differ in the group that makes up the C-terminus. So their sequence is basically tyrosine, glycine, glycine, phenylalanine, and then methionine or leucine. So they differ in the last amino acid. Now, the important part of the peptide is basically the tyrosine and the phenylalanine. So that's what they do. Now, and they function similarly to morphine. You know, morphine, the addictive substance. So they have a similar structure right here. This is the tyrosine part of the molecule. Now, there are two other molecules, and these are hormones. One of them is known as oxytocin, and the other one is known as vasopressin. Now, they have cyclic structures due to the formation of disulfide bond between two cysteines. Of course, you don't have to know the sequences, you just know the main characteristics. So you have the formation of disulfide bonds, so they are cyclic in nature, in structure. They also have amide groups at the C-terminus. So this is the amino terminus right here. Okay, that's amino acid number 1, 2, 3, etc. Now, to the carboxyl group at the end of the molecule of the peptide, you have an amide bond or amide group that is added. They both contain 9 residues, amino acid residues, except that oxytocin has isoleucine and leucine, vasopressin has phenylalanine and arginine, and that's what differentiates the two in terms of function. Where oxytocin is important in labor, muscle contraction during labor, that is during birth, So what it does is, so oxytocin is the hormone that induces contraction. Okay, so whenever contraction is induced in a mother in labor, oxytocin is given to this female. Now, vasopressin, on the other hand, plays a role in contracting smooth muscles in our gut. It increases water retention and it increases blood pressure as well. Now, this is the structure of vasopressin. Now, the reason why I placed this in here is not for you to memorize, but for you to practice. So I can bring you a certain stretch of amino acids or a certain peptide structure, and I can ask you what the amino acid sequence of this peptide is. So I have to recognize where the amino group is, okay, the start of the peptide, and you have to know the end as well. And you have to look at the amino acid at the R groups of the different amino acids. So right here, that's the first one, that's the cysteine. You look at the second one, that is a tyrosine. You look at the third one, that's the peptide bond. So you look at this one, this is phenylalanine. So again, another peptide bond right here. Okay, so you have to look at the amino acids one after the other. Okay, and recognize them. Now, two other peptides are known as gramicidin S and tyrosine A. Now, these two are cyclic decapeptides, meaning that they are made of 10 amino acids. They are bacterial proteins. They act as antibiotics. And an important feature of these two peptides is that they contain D and L amino acids. Now, usually, naturally, amino acids are in the L form in our bodies, but bacteria always have exceptions. Now, they also have ornithine as an amino acid, and ornithine sort of looks more like arginine, or it's a derivative of arginine, and it doesn't naturally exist in peptides. But in case of these two peptides, it does exist. Now, another important peptide is known as aspartame. Aspartame is an artificial sweetener, and it's used as a substitute for sugar. Now, aspartame is made of two amino acids, and these are known as aspartate and phenylalanine. Now, one important feature is that it has methanol at the end, at the carboxy terminus of the dipeptide. Now, now what's really interesting is that if you replace any one of these two amino acids by the D stereoisomer rather than the L, it would have a bitter taste rather than a sweet taste. Now, an important note about aspartame is that it should not be given to individuals with a condition known as phenylketonuria. Now, phenylketonuria is basically a hereditary inborn error of metabolism, and it's caused by a defective enzyme known as phenylalanine hydroxylase. Now, what phenylalanine hydroxylase does is that it converts phenylalanine to tyrosine as part of its metabolism. Now, people having defective phenylalanine hydroxylase cannot do this reaction, and as a result, phenylalanine accumulates, and it gets converted to another molecule known as phenylpyruvic acid. Okay. And phenylpyruvic acid accumulates in the central nervous system, and it causes mental retardation. And that's why people with phenylketonuria should not be given or should not ingest aspartame, because it increases the level of phenylalanine in their system. Okay, and phenylketonuria is one of these conditions that are tested for newly born individuals, babies, basically, to make sure that they don't have this condition, because it can be prevented simply by not giving them phenylalanine. Now, as a result, instead of having phenylalanine in aspartame, it has been replaced by alanine, okay, and the product is known as allitame. So this is sort of like an alternative for aspartame.